Welcome to this lecture on biotechnology and its application. What we're going to cover in this lecture is introduction. After that, principle of biotechnology, that how it works. Then tools of recombinant DNA technology. First, we will see what is biotechnology. Biotechnology, as the name suggests, is the mixture of biology with the technology. But how it defined? So the definition is it is the technique of using living organism or enzyme to produce different products which are helpful for the human beings. So one of the example is curd. It is produced with the help of Lactobacillus. It is a lactic acid producing bacteria. So the sour taste which occur in the curd, it is due to lactic acid production and it is produced by lactobacillus. Next example is the bread. As you can see over here, the dough size is continuously increasing with the time, right? It is because of the production of CO2 gas and it is produced by the yeast. Then the other example of wine. The yeast uses the grape sugar as a substrate and convert it as wine, or we can say the alcohol production. This process is also known as fermentation. Then the next application of biotechnology is IVF. It is known as in vitro fertilization. In vitro means it is also known as test tube baby. So the process is, first what we do is egg production is done with the help of hormonal therapy. After that we retrieve the egg in the petri plate and we collect the sperm sample. After that we allow the fertilization in one petri dish. After the fertilization, there is two cells stage. This is known as cleavage. One cell divide into two. Okay, over here you can see two cells. After that, four cell stage, then eight cell stage, 16 cell stage, which is also known as the morula. After that, the blastocyst production occur in case of blastula stage, and this blastocyst is transferred to the uterus. As you can see over here, this is the uterus and blastocyst is implanted into the uterus for the further development of baby or embryo. Next example is DNA vaccine. Nowadays, we use DNA for making vaccine in case of cancer and various other diseases. After that, correcting a defective gene, which is known as gene therapy. So these are the example we will see in detail in case of application. So first, there are two types of biotechnology or ways of biotechnology involved. First is genetic engineering and second one is bioprocess engineering. In case of genetic engineering, as the name suggests, we are something doing with the genetic material. So what we are doing? We are altering the DNA or RNA of the organism to change the phenotype of that organism. Then bioprocess engineering. Bioprocess, it means we are processing something to make certain products such as antibiotics, vaccine and enzyme. But what we are processing? So in this process what we do is we select some microorganisms, we grow them in the sterile condition or we can say uncontaminated condition where they produce certain enzymes or antibiotics and we purify them with down processing and use them for multiple purposes. Hope that's clear. So first we will see the basic difference of asexual and sexual reproduction as we are dealing with the genetic engineering. So in case of asexual reproduction, as you can see over here, only one parent is there. In case of sexual reproduction, there are two parents which are involved. Okay. So over here you can see the parent is completely identical to its progeny, or we can say vice versa, progeny is completely identical to the parent. But in case of sexual reproduction, there of mixing and matching, whereas mixing of genetic material takes place, whereas some genes of mother and some